focusing on the unraveling of the microenvironment in gastric MALT lymphoma and gastric cancer, if I've understood correctly. Please correct me if I get this wrong, Christy, but I'll, if everyone could please just mute their microphones, which I think everyone has done, and turn off their video just to minimize disruption while Christy's presentation is underway, that would be great. And with all of that, I will hand over to you, Christy, and let you share screen and start the presentation. Thank you.
Cool. Thank you. Thank you, Melissa, for your introduction. Do you see my screen?
Yes, that looks terrific. Good. Thank you.
Yeah. Yeah, thank you, Melissa, for the introduction, and also thank you for inviting me to talk here. So today I will go through a small journey with you using the multiplex immunohistochemistry technology. So we start with a bit of background. As many of us, we know that tumor microenvironment is a complex system. And because there's various cell types in it, you could see here, we can see in the tumor cells and various immune cells, including T cells, B cells, K cells, and tumor-associated macrophage, dendritic cells, etc. Oh, sorry. And those complicated system makes it heterogeneous tumor microenvironment, and this could potentially lead to the limited efficacy of cancer therapy. So now people are looking at which way we could improve the efficacy of cancer therapy, and one way is to do the cancer patient stratification. We could actually stratify patients with different factors, and I think tumor microenvironment is one of the most important factors that we need to look at. And with the cancer patient stratification, we could potentially enhance the cancer therapy, as well as we could predict patient prognosis. There's a lot of technologies that are available in the laboratories that could help us to do cancer patient stratification. And when we look back at the development of histology and pathology, we found several important milestones that also been widely applied, the technology widely applied in cancer research. One important milestone is the H&E staining that have been induced in 1870. We still nowadays still using it. And the other important milestone is in 1942, we introduced immunohistochemistry, and I believe a lot of researchers still using this technology nowadays. So with the development of highly multiplexed tissue imaging since 2013, which is around 10 years ago, we started to develop and using multiplex immunohistochemistry, and this is the most important part we would like to talk about today. So what is multiplex immunohistochemistry? I'll show it for multiplex IHC. So I have shown you two of the images that seem to like the multiplex immunofluorescence we have seen, but I was saying there's 5 markers in the human. On the top panel is a human colorectal cancer tissues. We have the pan-CK marker for tumor cells. We have PD-L1 labeling in green for the immune checkpoint markers, and CD8 for cytotoxic T cell markers, and FOXP3 for Treg cells, and DAPI for the nuclear staining. So I have applied this in the human tissues as well as in the mouse tissues. We could see there's several follicles of B cells, which is CD19 positive, showing in in green color, and also we could see in the mouse gastric tissues there's been infiltrated by dendritic cells and macrophages, which is positive for CD11C and F480. So with this background, I use this technology. So it's actually kind of multiplex immunofluorescence, but with different principle as the conventional IF. So the conventional IF we have been usually used in the laboratory is either we use direct immunofluorescence, which we use the primary antibody have already been conjugated with fluorophore. So with this method, the limitation is that there's no amplification of the signal. And the most common method we are now using is the indirect immunofluorescence staining, which we apply the labeled primary antibody first, then we followed by the secondary antibody with the fluorophore. So by using this indirect method, we could get the signal amplified. You could see the fluorophore in the yellow color. We actually see there's more signal in here when compared to the first direct immunofluorescence. However, quite differently, the multiplex IHC technology, it also has unlabeled primary antibody, but with a secondary antibody that has HRP conjugated here. And we also have a third terramine reagent, which also conjugated with fluorophore. So the HRP could help convert the inactivated terraform reagent into activated version. Then it allows them to covalently bind to the tyrosine residence near or at the antigen. This makes it give it more signal amplification when compared to the conventional immunofluorescence staining. And also it allows more biomarkers, because for the indirect RF staining, we have the limited antibody options because we need to use different antibodies derived from different species, while for the multiplex IHC, we could use the antibody raised from the same species for many times. So what's the advantage of this technology? So when we compare to the conventional IHC, we know that it only gives us one single marker per slide, which is limited information. And nowadays, more and more people are using flow cytometry. It is a fantastic technology, and I know that it could now we use detect more than 40 biomarkers at one time. However, the limitation is it's only limited to fresh samples. Especially in cancer research, we maybe want to do some retrospective study. For example, we are studying immune checkpoints and some immune markers now, but maybe if several years later we are interested in the other biomarkers, and if we want to go back and revisit our samples, and we need to correct new samples for our flow cytometry, while for the staining, so we could still use a paraffin embedded blocks to do more in studies. And also for flow cytometry, another limitation is that there's no spatial information, especially, there's kind of crosstalk and communication between different immune cells. So we could not really get those spatial information from the flow cytometry analysis. For the multiplex IHC, we could now able to stain up to 8 biomarkers on one formalin fixed paraffin embedded tissue section. And with the terramine signal amplification technology, we could detect targets with very low abundance. And also it could provide us more spatial information. So there's actually several commercial kits available from different companies. They all use the key technology for mlHC is terramine signaling amplification. And I have been using this technology since 2016, and I will use Opal staining kit as an example to explain how mlHC work and how I applied it in my cancer research. So how does the Opal kit staining work? So it also works as the other staining step is that we divide it into 3 steps. The first step is staining on slides, and the second step is we do imaging, and the last step is we do the analysis. For the staining, it's actually work quite differently as the conventional immunofluorescence staining. So when we get our slides prepared, we use the microwaving to do the epitope retrieval. Then we undergo several cycles for each antibody staining. We start with blocking step, and then we incubate the primary antibody, followed by the Opal polymer HRP reagent incubation, and then we use the TSA reagent, which gives the signal amplification. And after this staining, the most important steps is we will apply microwaving again to strip the unstained and antibody. So we go this round for one antibody, and we could repeat this circle for several times until we finish staining all the antibodies you want on the single slide. And then later on, in the last step, we could stain DAPI for the nucleus. So for the imaging, so now currently the company called Akoya Bioscience, they have several options for us to choose. They have the 4 kits, 4 color, 7 color, and 9 color kit, which allows we stain on the single slide. We could do 4 colors, 7 colors, and 9 colors. And for the 4 colors kit, we actually could do the imaging, use the confocal microscope. While if we do staining up to 7 or 9 color 9 colors, then we could only use multispectral imaging facilities instead of confocal. And if you are doing a lot of slides, we could also have the slide scanners to help to visualize the results. And when we go to do the analysis, there's several softwares that's been available. And one software is from the company called Inform. With the Inform software, we could do the spectral mixing to get the each biomarker staining results. And we could do cell segmentation to let us know which part is the tumor cells and the others is stroma cells, or what other cell segmentation. And the third part is cell phenotyping. With the biomarkers we have stained, we were able to identify the cells into different phenotypes. And with all this information ready in hand, we could further do more analysis using spatial signatures, but that will be doing in different softwares. So, and we also could apply third-party software, for example, QPath, to do more analysis. But I won't give too much detail on those details. But with this help of multiplex immunohistochemistry staining, we were able to visualize the tumor microenvironment in the cancer, and we quantify the expression of different biomarkers, identify different cell populations, and we could do more cancer patient stratifications and predict prognosis. So today I will give you some brief update on how I use and apply the mlHC technology. So I have used it to stratify cancer patients, and also I used it in a mouse model that to understand how it resembled the characteristic of the human gastric MALT lymphoma. And in the end, I will also quickly, briefly update a new technology in the multiplex IHC field. So start with a bit background on gastric cancer. As you may know, that gastric cancer is a fourth leading cause of cancer death, and the 5 survival rate is only 33%. And this the etiology for gastric cancer, I think there's a lot of factors being involved, including the bacteria called Helicobacter that colonize in the human stomach, and also there's a kind of factors including diet and genetic factors. There are several subtypes of gastric cancer. However, 80 to 90% of gastric cancer are adenocarcinoma, and some of the cancers are mucosa-associated lymphoid tissue lymphoma. We call it short for MALT lymphoma, and we also have gastric sarcoma and etc. The other small rare gastric cancer. So today I will mainly focus on the adenocarcinoma and the MALT lymphoma. So as you may be aware that the tumor microenvironment in cancer gastric cancer is also very complicated, increasing those classical immune cells. And also recently, you may be aware the immune checkpoints, the programmed cell death protein 1, short for PD-L1. So the it has the ligands of called PD-L1 and PD-L2. And these ligands are mainly expressed on tumor cells. So the binding of the PD-1 and the PD-L1 will lead to the inhibition of T cell activation and proliferation. So we are curious about whether we could study and investigate those immune checkpoint markers as well as those classical immune cell markers in the tumor microenvironments that maybe could potentially help us to do patient stratification and help us to extract some valuable prognostic markers. So with these potential questions, we are lucky enough that we could get some gastric cancer patient samples. So we harvested 90 paired tumor and adjacent normal tissues from gastric adenocarcinoma patients from our collaborator in China. So we are able to make a tissue microarrays that have those 90 paired tumor and adjacent normal tissues on one slide. And we did a H&E staining to examine how the samples look like, and due to the incompleteness of some tissues, we only able to get 84 paired samples for downstream analysis. So after the H&E staining evaluation, we started to do the Opal follicular staining, and we want to look at some markers in the gastric cancer microenvironment. So this work was actually done a while ago during my PhD study, and the 4 color kits I'm using is I'm using the PD-L1, using the Opal 520. DAPI is a nucleus marker, and I use the FOXP3, which is a marker for Treg, and it's using Opal 570, and CD8 is using Opal 670. So because as all of the work was done at Hudson, and we actually do not have the high spectrum imaging system at Monash. So, and also it's because it's just a 4 color staining. So I managed to use the confocal microscope at Hudson to visualize the results. So this is a merged image showing the staining of PD-1, CD8, and FOXP3. And also we could see the single staining of the individual markers, and we could see the staining is showing very specific pattern, and there was like very little background. So with the optimization of this, and we staining the whole tissue microarray, and I could show you some represented images from each of the core. Then this is the 2 images from different patient, and we could already see the heterogeneity between different patients. So further on, we quantified the density of the FOXP3 positive cells, PD-L1 positive cells, and CD8 positive cells in both adjacent normal tissues as well as tumor tissues. We did find there's increased expression of FOXP3 positive Treg cells and PD-L1 positive cells in the tumor samples when compared to the adjacent normal, where we see the decreased expression of CD8 positive T cells in the tumor samples compared to the adjacent normal. We further calculate the ratio of CD8 to FOXP3 and CD8 to PD-L1, because when we're thinking about just evaluating in single marker manner, we want to look at the relative relationship between those markers. And interestingly, we did see a significant reduction of CD8 to FOXP3 ratio in tumor cells compared to adjacent normal. And later on, when we examine all the staining results, we did find there are several interesting subgroups of gastric cancer patients. We could see there's a patient with expression of PD-L1, the immune checkpoint marker, but with less immune cell marker, with less CD8 and FOXP3 expression in the tissues. We see the second pattern is the tissues have very high expression of PD-L1 and also very abundant expression of tumor infiltrating lymphocytes, especially CD8 and FOXP3. We also see the third pattern is less PD-L1 expression, but with abundant immune cell infiltration, while the fourth subtype is kind of like cold tumor, like the second type is kind of hot tumor, while the fourth part is like cold tumor, is that not too much immune cells and also not too much PD-L1 expression. We first want to use the ratio of CD8 to PD-L1 and CD8 to FOXP3 to do patient stratification. With the unsupervised hierarchical clustering, we were able to divide these 84 gastric cancer patients into 3 clusters. And when we look at the first clusters highlighted in the red box, we see those patients that with low CD8 to PD-L1 ratio, while moderate CD8 to FOXP3 ratios. We further look at the survival outcomes, and surprisingly, we found the patients with high PD-L1 expression in this subcluster showing much better survival outcomes compared to the PD-L1 low expression patient, which surprisingly we find the PD-L1 is a prognostic marker in the subgroup of the cancer patients. We further validate, we download the RNA sequencing data from TCGA portal, and we did the similar analysis as our mlHC data. And interestingly, we find similar results and as the mlHC results. So just to quickly summarize, in this story, I used the multiplex immunohistochemistry staining on tissue array, and we find the PD-L1 expression is a missing prognostic factors in a subgroup of gastric cancer. So to be noticed, if we just use PD-L1 as a marker for all the cancer patients, it did not really have those prognostic value, only in a subgroup of populations. I think that's the importance of the cancer patient stratification that allows us to identify certain subgroups of patients that we could know there's the prognostic factors. And also we are very excited to see it's very consistent that we could see the cluster of the patient in both our cohort that's been identified by the multiplex IHC, which is actually protein level, and this consistency with the TCGA RNA level. So this is kind of story for this. And further on, one of my collaborators also worked on the gastric cancer. We together looked at more using more markers. So in this another individual study, we have been using the gastric cancer tumor samples. We have partly used it for FFPE blocks to do the multiplex IHC staining to understand the density of the different immune cell populations, as well as we apply the distance analysis. We also get some RNA samples from those tumor samples and do some RNA microarray analysis. We were able to get some blood samples, and we isolate PBMC and also do cytoph to look at more PBMC phenotyping. And we combine all this information together and with the follow-up information from patients and further to characterize a promising prognostic marker from that. So for this story, we actually applied the Opal 7 kit because this work was done at PintMac, and my collaborator Mingyu Huang, she's the main people who did this work. And with the 7 kit, and also because PintMac has the vector multispectral imaging system, so we could do more analysis on the staining results. So we could see using these 7 markers, we were able to phenotype the cells into double negative T cells, which is CD3 positive, but CD4 and CD8 negative. And also we identify CD4 positive, which is CD3 and CD4 positive and CD8 positive cells. And also the Treg cells is CD4 positive and FOXP3 positive, and the CD56 positive for NK markers and also tumor cells. So with this background, we further did a distance analysis. We introduced a new algorithm that we could calculate the nearest distance between 2 cell type, and we could further look at the distance between all the cell types that we have been labeled in the slides. So this just to give you an example. So this is a distance to the CD4 and the FOXP3 double positive T cells. From the left panel in this patient, we could see the nearest cells, nearest distance to the Treg cells is tumor cells in this patient, while in the other patient, the nearest cells is CD8 positive T cells. So actually, it's also the distance between different immune cells are also heterogeneous, which makes it even more difficult for us to find more information. So that's why we need to get those information out by the multiplex IHC technology. And in that story, we're also able to identify some subgroups of patients. Later on, if we have the survival outcomes from patient, we were able to find one of the subpopulation, which was a high expression of CD8 and the high expression of CD4 and FOXP3. These patients is having better survival outcomes compared to the other group. We have get this validated also in the previous cohorts, that is 84 samples. So in this story, so we were able to integrate the multiplex IHC staining with other technologies that could used for us for gastric cancer patient stratification. And also we started to doing some simple spatial analysis. So another story, due to time, I will go too deep into it, just how I use it, because for years, a lot of people started to use this technology, and most of people are using this technology in human tissues. And there's not so many kits available in mouse tissues. And I started to work and optimize the condition in mouse tissues, and I successfully managed to do the multiplex IHC on mouse tissues. And at that time, we have a novel mouse model that could reproduce the key characteristics of tumor in human gastric B-cell lymphoma. And we could see from the H&E staining, those mouse tissues showing the same as human tissues, showing the lymphoid follicles across the gastric mucosa with the hallmark of lymphoid epithelial lesions, highlights here. So we further ask whether this mouse model could have the similar tumor microenvironment as human. So we applied various markers, and we found actually it is quite similar, and our mouse model could recapture the future feature of the human gastric B-cell lymphoma. And it's especially interesting, we could see there's abundant expression on the right-left corner. There is this abundant expression of PD-1 and PD-L1 expression in the gastric tissue. So I think this mouse model is will be very ideal for evaluating, the anti-PD-1 or PD-L1 antibodies in this disease. So again, I just quickly summarize. So this technology could now be applied in mouse tissues, and we could use more markers. Although it needs a bit of optimization, but we are now using more and more markers to put them together. So the last slide that I would like to share with you is a new technology that have been emerging recently. Is now people are starting to doing single cell spatial phenotyping, which allows more than 100 protein biomarkers on a single human FFPE tissue. And I give you just a very quick example. Is this here showing is a 103 plex panel for one slide. And we could see it has includes the classical immune cell markers and different lymphocytes, myeloid modules, and the activation, or even metabolic markers. So this makes us even like more information, and with those massive and abundant information, we could further do cross-correlation analysis, more spatial analysis, and co-localization, and more mechanistic studies into that. So just want to quickly summarize and give you some take-home messages. The first thing is we now know like multiplex IHC is a powerful tool to describe the tumor microenvironment. I think it could be used in most of the solid tumors, because it could provide the expression of various biomarkers as well as the spatial information. And it could also be effectively employed for both human and mouse tissues. And we also have successfully applied in tissue microarrays. It could also be combined with other technologies, which could help us to stratify patients and also potentially identify prognostic markers. And lastly, with a new technology, single cell spatial phenotyping, which will enable us more critical mechanistic insights into cancer cell biology. Yeah, so that's all from me. And lastly, I want to acknowledge all my supervisors from since my PhD study and my postdoc, Professor Brian Williams, Professor Richard Ferraro, and Paul Hudson, and all the other people from Hudson, and my the platform, histology platform, and micro imaging platform from Monash, and also our collaborators providing those precious clinical samples. And thank you for listening, and I'm happy to take any question.
Hey, thanks so much, Christy. Wonderful pieces of artwork at the end.
Thank you.
They're just lovely. They're the sorts of things you see put up on posters in competitions. That was lovely. But questions for Christy?
Yeah. Do I still need to share the slide, or just it's fine?
I think this is fine, unless we need to go back to reference something.
Yeah.
Perhaps I could start off if there's no one me. Oh no, here we go, Hannah. Oh, clap your hands. Okay. Right. Christy, the wonderful capacity to do things spatially and identify cells that have particular features from others, I guess from a molecular minded person, in addition to protein, have people been using those sorts of stained sections to laser capture material for ongoing molecular analyses? Is that possible?
Yeah, so one of my ongoing work is doing some markers like related to molecular biology. I did, because it's ongoing work. So yeah, so I try to introduce more markers into this panel, and then we could further look at the co-localization as well as how it worked in the certain pathways. So I think it's potential, but I guess the only concern is that for every time when we introduce new antibodies, we also need to always need to optimize the condition for the staining, and how it could combine into the multiplex staining, because we also need to think about the relationship with other markers. Yeah.
I think that came into my mind particularly when you referred to hot and cold.
Yeah.
Immuno,
Yeah.
which of course from somatic geneticist point of view, we use that language as well.
Yeah.
But it means something a bit different, I think, but presumably it's actually identifying something similar. Do you know if those ones that you're calling hot as far as the immunohistochemical response are actually hot in terms of a large number of somatic genetic changes in the tumour, or is it something completely different?
I think it's a kind of different concept, because when I think hot and cold tumor in my presentation, I think it's more kind of I will identify hot tumor as, there's a lot of immune cell infiltration in the tumor microbiome, in the tumor microenvironment, as well as the abundant expression of different immune checkpoint markers. So we think those kind of tumor microenvironment is kind of hot tumor, which usually those samples will, patients will have better chance to respond safe to the immune checkpoint therapy or other therapy. While for the cold tumors, usually they do not really express, these immune checkpoints and with less immune cell infiltration. So it's kind of a bit different, I guess.
But have you done the work? Have you actually looked to see if the two are there? You don't often extend your work into doing somatic genome analysis?
No, we haven't done so, but I think that's a good point that we could do. Yeah.
Be interesting. I think often there are scenarios where, a lot of somatic variation acquired by a tumor can actually provoke an immune response. And there may not be the same pathways, but I think they may be in concert and might be interesting to look.
Yeah.
Okay, so a question in the chat about how much is the average cost for staining one mlHC with such 100+ protein markers? How long does it take to generate? It's a very practical question, but I'm sure you could comment.
I would say, generally it's not cheap. It's kind of expensive. I don't have the actual number in my mind currently, but we could ask company because I think there are several companies that could provide those service. And I think for the times, because the technology, how they achieve more than 100 biomarkers on one slide, is that they could actually staining, maybe 7 or 8 markers at one time, and then they strip off the antibody, and then they reintroduce new panels of antibody, and then go several rounds and up to 100 markers. So I guess that will take around 10.5 months to finish that process. But for the analysis part, it will take much longer time. And for the cost, sorry, I couldn't give you very accurate information. Maybe we could inquire company if you are interested.
Can you give a ballpark? Like how many zeros? 2, 3, 5?
I think several zeros.
Several zeros. Okay, and a question from Kevin. Would liquid biopsy complement the stratification of patient cohort given it's less invasive?
I don't know. It's kind of difficult to conclude because for liquid biopsies, I think, for example, like blood or PBMCs, I think the scenario may be quite different as the solid tumors. I think it's difficult to say because I don't really have too much knowledge on the liquid biopsies. But I think for the multiplex IHC, which gives us the in situ information, then we could consider the expression and the spatial information, which will give more kind of accurate results compared, to the liquid biopsies.
But it would theoretically work on isolated circulating tumor cells or something, presumably, wouldn't it, if they were?
Yeah, I think.
Fixed?
Yeah, I think potentially yes.
Okay. Zhang Ning is a person of similar mind to me. Can this technique be combined with spatial transcriptomics? I went genetics. This is transcriptomics.
I think potentially yes, but I think the question will be how could we combine? Because I know there's the single cells spatial transcriptomics from the other companies that we also be able to identify more protein biomarkers. I think there's kind of potential if we could have the slides washed off and then do another spatial transcription on the same slides, then we could maybe achieve that.
Terrific. Okay, are there any other questions though, Christy, just at the moment? If not, Christy, we know where you are. If we had more questions, I guess we can follow up via email or something in the future.
Yeah. So maybe I'll have my email.
I think if I think it's really just left to thank you again for a really terrific technical demonstration of what multiplex immunohistochemistry can do in certain contexts, and help us explore what else it might do in extending into the work of others. It was really always a joy to see histopathology presented as the baseline of people's consideration of cancer. I know it's not always possible. It's always a great joy to see that in such beautiful colour as well. So thank you, Christy, and thanks everyone for attending. And the future tech talks will be available via the usual channels and will be circulated via the usual channels for future talks. So I hope to see some of you again then.
Okay.
So thank you.
See you. Bye.